Soupli from the French National Center for Scientific Research and Genoscreen, and he's talking about the present and the future. Deeplex-MycTB, an all-in-one test for diagnosis and surveillance of drug-resistant tuberculosis. Philip.
Thank you, Daniela, and thank you for giving me the opportunity to present the work that we have done to develop this new test for early diagnosis and surveillance of drug-resistant TB. According to the last WHO TB report, there is an urgent need to close the gap that remains for diagnosing hundreds of thousands of MDR-TB cases. Phenotypic testing is too slow, and the current molecular tests only interrogate common mutations in relatively few targets. So as we had the chance to show together with Tim Peters and Derek Crook's team in Oxford, whole genome sequencing holds great promise to overcome these limitations, as it can efficiently predict drug resistance based on whole genome sequencing. But at the same time, it could be observed in this study that all the determinants that were identified as resistant determinants were concentrated in only 14 candidate genes for the drugs that were considered in this study, opening the way for a targeted assay as an alternative. So this kind of approach, whole genome sequencing, was used and evaluated prospectively for complete microbacterial diagnostics as an alternative to classical workflows. And it was shown that indeed whole genome sequencing was able to predict a species and drug susceptibility with high accuracy and in a way that was faster and cheaper compared to classical diagnostics. But still, for performing this, you need a primary culture. This delays diagnostic by 1 week or more in many cases. Technically, in principle, whole genome sequencing can be done on clinical samples directly, but at a very low multiplexing rate with low coverage depth, or by using costly capture systems and laborious capture systems. So therefore, we need something else. Thus, at Genoscreen, we have developed a new test, which is called Deeplex-MycTB, that can be used directly on clinical samples. So this test consists of a targeted next-generation sequencing assay of a single 24-plex amplification of main resistance targets plus species identification and MTBC strain genotyping targets, which makes it all-in-one design. So this targeted approach allows for deep sequencing of the samples for sensitive detection of heteroresistance, and at the same time, parallel and cost-effective analysis of up to 50 samples in a single MiSeq run, or up to 70 samples per a single MinSeq run. And importantly as well, this molecular test is coupled to a fast and easy-to-use application for easy analysis of the NGS data that is hosted on a highly secured and high-performance cloud. This test includes 18 targets associated to resistance to first-line and most second-line drugs, including all the significant targets that were identified in Walker's study, plus other targets associated to drugs that were not included in this study, including the target that is largely associated with resistance to bedaquiline, the newest available drug for treating TB. So on top of that, we use HSP65 in combination with ribosomal DNA targets for microbacterial species identification, and for MTBC strain genotyping, we use the target that is used for oligotyping in combination with phylogenetic SNPs that can be found in all the targets associated to drug resistance, plus eventually the mutations that are associated with resistance for further guiding the molecular surveillance investigation. And we have, in addition to that, an internal DNA control to control for the presence of PCR inhibitors in clinical samples, which is important. So the Duplex test relies on a calibrated pipeline which comprises 4 main compartments. One comprises the compartment for species identification, which uses published databases which comprises more than 140 reference sequences from different mycobacterial species. For oligotyping and phylogenetic SNPs, we used the CivVit-Web database that corresponds to the largest polygotype database available worldwide. And for phylogenetic SNPs, we use the FIS-RESI data as published by Fourier, Riegel, and colleagues in 2014. For interpreting the mutations in terms of drug-resistant association or lack of resistance, we use different reference databases, such as FIRES-E, plus the one that was published with Walker and colleagues, plus for PNCA, the one that was published by Mioto and colleagues recently, and when it will become available, we will include, of course, ResectTB and even cryptic data. So, and we even have here a metagenomics compartment, which gives indications about the presence of other bacterial species than mycobacteria that could be present in the sample, but it is important to note here that this is indicative because, of course, the whole test is biased towards detection of mycobacteria for obvious reasons. So regarding the coverage depth that we can achieve, so this is the range of coverage depth that we can achieve across different numbers of genome equivalent in the PCR reaction, and you can see that we have very high coverage depths that can be achieved over a wide range of DNA concentration that cover, of course, the range that can be achieved by microscopic detection, but we can even go below, down to 100 and even 10 genomes with relatively large coverage depths of several hundreds, even more than 1,000 under these conditions. So based on this high sensitivity of PCR, with the, of course, the taking into account the DNA extraction step, which is critical, and the bacterial load, we can even analyze part of the smear negative samples, such as in this example, showing here a full match of the H6P65 sequence obtained for this sample over the complete read set virtually, so indicating that here we have an MTB, a strain in the sample, which is confirmed by the metagenomics analysis. Here you have the coverage depths again over all the different targets in this smear negative sample with very high coverage depths. And here you have the result of oligotype identification. In this case, we have a very common oligotype, which is ST53, part of the Euro-American lineage, a very classical oligotype. So regarding the variant detection level that we can obtain, so we have calibrated here this variant detection level by analyzing, for instance, large set of drug-susceptible and multi-drug resistant isolates plus internal controls. And this shows you here the level of variation that was achieved by over more than 200 samples. It's accumulated results over the different positions of one amplicon corresponding to RPOB. You can see here all the samples, all the cases where mutations were detected and fixed in the samples. Here we have the RDR hotspot, where most of the mutations are concentrated, but you can see we cover other regions of RPOB that are not included in the expert test, but can be very frequently found in MDR strains, for instance, in South Africa, as recently reported. So you have here a zoom in of the background variation that is detected by the assay, and you can see here that this background variation is below 2%, except in one position specifically here for this target, but we don't exceed 3% in this case, and even so this defines the maximal level of sensitivity that we can achieve, and we believe that it will be likely sufficient in many cases, as the 1% fraction of phenotypically resistant population that is considered for DST, classical DST, might in fact be an underestimate in many cases, because resistance is associated often with a fitness cost that counter-select resistant strains in culture. So we have as well evaluated the capacity of the test to semi-quantitatively measure the proportions of wild type versus mutant population in samples. We obtain a linear curve here, and we can go down even to close to 1%. which is above the level of noise that is detected with a drug-susceptible mixture. So we have a semi-quantitative detection of levels that are close to 1% there. If we go below 1%, we become very close from the noise, so we prefer to stay above 2% for this reason. So regarding the ongoing evaluation of the test, so our in-house evaluation on clinical specimens showed that the test is effective on most largely the large majority of the smear-negative samples, and about 25% of the smear-negative culture-positive specimens as well. So we are evaluating now the sensitivity and specificity regarding detection of drug resistance of the test using the WHO TDR MTBC strain bank, which is very useful because it covers a large diversity of phenotypic and genotypic profiles, genetic resistant profiles and different geographic origins. And this collection was used previously to evaluate the Cepheid GeneXpert MTB RIF test, which is a useful benchmark for us. And we have ongoing beta testing of the test in collaboration with Daniela in Milano, and as well with Bouke de Jong in Antwerp, where we analyze a large set of clinical samples coming from 2 WHO drug resistance surveys organized in Djibouti and DRC. And I will show you preliminary data for the Djibouti collection that we have analyzed at GenoScreen using samples that were sent by Antwerp to us. So importantly, the analysis was done using just the equivalent of 100 microliters of clinical sample versus 500 or 1,000 microliters that is usually used with the expert test, as done in Kinshasa. So despite this low amount of sample at the beginning, we were able to obtain exploitable results for more than 90% of the samples with sufficient on-target coverage. And it should be mentioned here that we even have a higher proportion of invalid samples when analyzed by the Hein test. So based on this, we could predict 15 multi-drug resistant TB cases in that collection, which is maybe in the range that could be expected in this region. So, and among the 363 samples that were detected and identified as RIF sensitive by the expert test, we only found one discordant case with our Duplex assay. And in that case, we detected a double mutation in the sample that was concerned. So that leaves us with little doubt that this result is true. So, and among the 36 samples that were identified as RIF resistant by the expert, we confirmed 27 of them, but we identify 7 that were RIF sensitive, predicted as RIF sensitive by using our Duplex assays, and 2 with mutations, but with conflictive prediction depending on the allele-calling database that we consider for prediction. And these 7 RIF sensitive cases, as identified by the Duplex assay, were all confirmed by single sequencing and Hein test, which is looking very, very good. So regarding the deployment of the test, so we plan deployment on the cloud by early 2017, and this deployment of the application on the cloud